We'll start. Yes. Okay. Let me share. Yesterday we have finished the dicot stem. Today, today's topic is this is monocot TS of monocot stem. The internal structure of TS monocot stem shows four parts, not like dicot root or dicot stem. TS of monocot stem best example is Gia maize. The common name is maize. This is the maize plant. This is female inflorescence. This is male inflorescence. This is root system. This is the stem. If you see the cross section of the stem, so it is this is the ground plan. So this is ground plan. So this is stem. If you take the TS, the transverse section is like this. This is the vascular bundle. If you have a look, close look, the vascular bundle appears like this. So yeah, TS of monocot stem shows epidermis, cortex, ground tissue, vascular bundles. Epidermis is the outermost layer. It is made up of single layer of cells. So this is the epidermis. This is epidermis. This is ground tissue. This is ground tissue. Just below the epidermis, you can find hypodermis. These are vascular bundles. These are vascular bundles. If you see the TS of monocot stem under the microscope, compound microscope, it appears like this. Here you can see the tissues very clearly. So epidermis is made up of single layer of cells. Cells are barrel shaped. And it is one more important thing in epidermis. It is very easily to recognize, very easy to recognize. The epidermis is covered with thick cuticle. Comparatively in dicots, cuticle is thin. In monocots, cuticle layer is very thick. Yesterday we have seen trichomes were present. Trichomes were present. in dicots. Whereas here you cannot see any epidermal hairs. So this is the difference between dicot stem and monocot stem. In monocot stem, epidermis is as usual. It is single layered membrane or layer. Cells are barrel shaped, closely packed. You can see there is no secondary growth. That's why you can see stomata also. But the difference between dicot root and dicot stem and monocot stem, where cuticle layer is thin in dicot stem, whereas in monocot stem, cuticle layer is thick. Thick cuticle layer is present in monocot stem. And trichomes are also absent. There are no trichomes. Trichomes are nothing but they are epidermal hairs. They are absent. And the second part of transverse section of monocot stem is cortex. This is cortex. See, 2 to 3 layered or 4 layered cells are seen just below the epidermis. This is cortex. Here cortex is reduced or poorly developed. It is restricted to hypodermis only. No general cortex and no endodermis. These both the parts are absent in monocot stem. I'll repeat once again. Cortex in monocot stem is restricted to hypodermis only. General cortex and endodermis are absent. General cortex, that is middle cortex, and innermost layer, that is endodermis, is absent in monocot stem. Hypodermis generally is sclerenchymatous. Sclerenchymatous hypodermis is present in monocot stem. Whereas in dicot stem, the hypodermis, that is outer cortex, is cholenchymatous. Yesterday we have seen. So in this sclerenchymatous, hypodermis is present in monocot stem. It is 2 to 3 layered cells are there. It provides, since hypodermis is made up of dead cells, that is sclerenchymatous cells, it provides exclusively mechanical support. And the next part is large. It occupies large parenchymatous tissue, the central column. These are the cells which are in different sizes and in different shapes. This is normal parenchymatous tissue. This is called ground tissue. This is the third part of monocot stem. Ground tissue. It is simple parenchymatous tissue. Cells are in different sizes and shapes. Cells are loosely packed with small intercellular spaces. Generally, it stores food materials and water. I'll repeat once again. Ground tissue occupies large space. The central column of the stem is having a simple parenchymatous tissue. It is a living tissue. Generally, the cells are in different sizes and shapes. They are closely or loosely packed, small intercellular spaces. The main role of ground tissue is storage. It stores food materials and water and different substances. Here you can see there are vascular bundles could be seen in the ground tissue. There are many, as you have seen already in ground plan. Let us have a look once again. This is the ground plan. These are the vascular bundles. A group of vascular tissues are there. These are vascular bundles. The number of vascular bundles are more than 20 or 30. Many number of vascular bundles are there. Numerous vascular bundles are there in monocot stem. Comparatively in dicot, there is a limited number of vascular bundles were there yesterday. We have seen in the case of dicot stem. If you see the monocot stem like maize stem, the number of vascular bundles are numerous, countless, more in number. And there is no, the arrangement is irregular. You can see they are scattered irregularly. The vascular bundles are scattered irregularly. Such type of arrangement of vascular bundles in steel is called attack to steel. Attack to steel. I'll repeat once again. What do you mean by attack to steel? Attack to steel is a steel in which the numerous vascular bundles are scattered irregularly in the ground tissue. I'll repeat once again. In monocot stem, the type of steel is attack to steel. The type of steel is attack to steel. What do you mean by attack to steel? Attack to steel is a steel where numerous vascular bundles are scattered irregularly. There is no particular arrangement. There is no particular sequence. And the sizes of vascular bundles are also different. Some are smaller and some are bigger, larger. You can see irregular arrangement of vascular bundles scattered in the ground tissue. Such type of arrangement of vascular bundles in ground tissue is called attack to steel. Attack to steel. See, the sizes here you can see this vascular bundle is smaller. These are larger comparatively. They are larger in size. This is smaller vascular bundles. So let us go in detail. As I told you, the TS of monocot stem shows 4 parts: epidermis, hypodermis, ground tissue, vascular bundles. So this is epidermis, a thick layer of outermost cells. You can see they are closely packed, they are barrel shaped, closely packed, barrel shaped, without intercellular spaces. This is the outermost layer of monocot stem. Generally, the epidermal cells are parenchymatous, without intercellular spaces and without chloroplast. Except in God cells of stomata, the epidermal cells, chloroplasts are absent. Chloroplasts are absent. The outer surface of the epidermis is covered with thick cuticle. Thick cuticle. And there are no trichomes. Epidermal hairs are absent in monocot stem. This is the prominent feature of monocot stem. Thick cuticle, absence of trichomes. This is the first part. The next one is cortex. As I said, here you can see just below the epidermis, you can see a few layered thick cells, closely packed cells are seen. This is hypodermis. In monocot stem, cortex is restricted to or reduced to hypodermis only. General cortex or middle cortex and endodermis are absent. In monocot stem, cortex is reduced. It is limited to hypodermis. The hypodermis in monocot stem is made up of sclerenchymatous cells. It is a sclerenchyma tissue. As you know, sclerenchyma is a dead tissue. It provides mechanical support. It provides mechanical support. In some sense, the hypodermis may be also composed of few chlorenchymatous cells. Here in the figure, you can see the patches of chlorenchyma. Patches of chlorenchyma. It is rare cases. In some of the monocot stems, you can find chlorenchyma patches in hypodermis. It helps in photosynthesis. It helps in photosynthesis. And the main role of hypodermis is it provides mechanical support. Provides mechanical support. Next one, ground tissue. This is the third part of monocot stem. Here you can see the oval or spherical, irregularly arranged cells with small intercellular spaces. This tissue is a simple parenchymatous tissue. In monocot stem, just below the hypodermis, a large central column is composed of simple parenchymatous tissue. They are spherical or polygonal or oval in shape. They are of different sizes. Some of them are very smaller and some of them are very bigger, larger. They have intercellular spaces, empty spaces left over between the cells. The main role of this tissue is, ground tissue is storage. It stores food materials. In monocot stem, no general cortex, no endodermis. Only it is restricted to sclerenchymatous hypodermis. Not like dicot stem. Stele is here attacked to stele. Here you can see the number of vascular bundles are numerous. They are in different sizes. They are in different sizes and scattered irregularly. Scattered irregularly. Such type of stele is called atactostele. In monocot stem, the identification feature of monocot stem is atactostele. The presence of atactostele is the main identification feature. Identification feature. Here, in monocot stem, pericycle, medulla, and medullary rays are absent. In cortex, general cortex and endodermis are absent. In cortex, general cortex and endodermis are absent. In stele, pericycle, medulla, and medullary rays are absent. Only the scattered numerous vascular bundles could be seen, which are scattered irregularly in the ground tissue. Ground tissue. Such type of arrangement of vascular bundles in monocot stem is called atactostele. Atactostele. The next one is vascular bundles. So this is individual vascular bundle. Each vascular bundle is like this. Generally, in monocot stem, the vascular bundles which are arranged towards the periphery, that means peripheral vascular bundles are smaller in size comparatively. They are smaller in size. The vascular bundles which are arranged towards the center are larger. The vascular bundles, the peripheral vascular bundles are smaller and central vascular bundles are larger. And each vascular bundle is enclosed with sclerenchymatous bundle sheath. These cells are sclerenchyma cells. It forms like a sheath around the vascular bundle. That means each vascular bundle is enclosed with sclerenchymatous tissue. This is generally known as bundle sheath. Generally, this sclerenchymal tissue is present around the vascular tissues in monocot stem as a bundle sheath. Since it is made up of sclerenchyma, it provides mechanical support. It provides mechanical support to the vascular tissues like xylem and phloem. In monocot stem, vascular bundles are conjoined. You know what do you mean by conjoined? Both here, this is the xylem patch. This is the xylem patch. These are the phloem patch. These are the xylem vessels. This is phloem. This is xylem. The large empty spaces could be seen here. These are xylem vessels. These are two are metaxylem vessels. This is protoxylem vessel. Generally, in each vascular bundle, phloem and xylem are together, are side by side. If they are together, then the term is used conjoined. If they are on the same radius side by side, then the term is used to describe such vascular bundle is collateral. I'll repeat. The vascular bundles in monocot stem are conjoined, collateral, closed type of vascular bundles are there. You know what do you mean by conjoined? Conjoined means the number of vascular bundles are numerous, arranged irregularly, are scattered in the ground tissue. Such type is called attractive state. And if you observe closely, each vascular bundle is oval in shape, enclosed with sclerenchymatous bundle sheath. It is having few xylem elements and few phloem elements. This is phloem patch. These are xylem vessels. Xylem vessels. Xylem and phloem are together, hence the name conjoined. If they are side by side on the same radius, then the term is used collateral. If you observe closely, there is no cambium, vascular cambium. There is no vascular meristematic tissue in monocot vascular bundle. That's why this type of vascular bundle is called closed vascular bundle. Whereas in dicot stem, the xylem and phloem are separated by a strip of vascular cambium. That is, meristematic tissue is present between xylem and phloem in dicot stem. Vascular bundles of dicot stem. Such type of vascular bundles in dicot stem are described as open type of vascular bundles. Whereas in monocot stem, the vascular bundles are closed. They are closed type of vascular bundles. Since there is no vascular cambium, which is separating in xylem and phloem. In dicot stem, a strip of cambium is there. That's why in dicot stem, they are open type of vascular bundles. But in monocot stem, such vascular cambium is absent. That's why these vascular bundles are described as closed type of vascular bundles. So the presence of atactostele is the main feature of monocot stem. And the vascular bundles are conjoined, collateral, closed type of vascular bundles are there. If you observe the xylem tissue in each vascular bundle, you can have a look here. So this is phloem. These are the xylem. Metaxylem vessels. These metaxylem vessels are larger in size. This is protoxylem. The central xylem vessel is called protoxylem. Each vascular bundle appears like this. This dark color tissue is sclerenchymatous bundle sheath. This light blue color cells are phloem cells, phloem elements, and this round empty spaces are xylem vessels. These two are which are larger in size comparatively. Such vessels are called metaxylem elements. And this is the central, a single vessel is seen, which is smaller than the metaxylem elements, is called protoxylem. This is the first formed xylem. And there is a, adjacent to the protoxylem, there is a large air space is seen. This air space is formed due to disintegration of protoxylem. That means in the initial stage, there are two metaxylem elements and two protoxylem elements were seen. At later stages in the vascular bundle, one of the protoxylem element disintegrating and leaving a large airspace. This airspace is formed due to disintegration of protoxylem. That's why this cavity, this airspace is called lysigenous cavity. Lysigenous cavity. What do you mean by lysigenous cavity? Due to disintegration of this protoxylem, this cavity is formed. This generally is filled with air or water. Air or sometimes water may be also seen in the space which is formed after the disintegration of protoxylem. That's why this is called lysigenous cavity, or else it is formed due to protoxylem disintegration. That's why it is also called as protoxylem lacunae. Lacunae means empty space. Cavity is formed due to disintegration of protoxylem. That's why this is called protoxylem lacunae or lysigenous cavity or air space. This is seen in each vascular bundle of monocot stem. In each vascular bundle, there is a air space adjacent to the protoxylem. That is air space or lysigenous cavity or protoxylem lacunae. This is about xylem. And there is a phloem. Phloem, in each vascular bundle, there is a patch of phloem which is always towards outside of the stem in each vascular bundle. The inner side, you can see the protoxylem. That's why this condition is called endark. Endark xylem is there in stems. Exarch xylem is present in roots. Like dicot stem, in monocot stem also, protoxylem is towards the center. That's why the condition router was off. Uh, that's why the connection is lost. Let me continue. Let me continue. In xylem, there are two metaxylem elements are there and one protoxylem element is there. One of the protoxylem is disintegrated, forming protoxylem lacuna or lysigenous cavity or air space. This is formed due to disintegration of one of the protoxylem. And the protoxylem is always toward the inner side of the vascular bundle, towards the center of the stem. That's why that condition is called endar. That condition is termed as endar. If you see the phloem, between the two metaxylem elements, there is a patch of phloem is present. In each mature vascular bundle, few elements, sieve elements and the companion cells could be seen as a patch. It is present between the two metaxylem elements. You know the function of phloem? It transports food materials. It transports food materials. So this is the summary of the monocot stem. Here, as I said earlier in this class, the TS of monocot stem is having four parts. The first part is epidermis, the second is hypodermis, the third one is ground tissue, and the fourth part is vascular bundles. The epidermis is the outermost layer. It gives protection. The external surface of the epidermis is covered with thick cuticle, and trichomes are absent here. That you need to remember. The presence of thick cuticle and absence of trichomes is the key feature, identification feature of monocot stem. And the second part of monocot stem is cortex. Here cortex is restricted to hypodermis only. No general cortex, no endodermis. These both are absent in monocot stem. Only sclerenchymatous hypodermis is present. It gives only mechanical support. It provides mechanical support. And the vascular bundles are numerous. In monocot stem, the number of vascular bundles are countless, numerous, and they are irregularly arranged and scattered in the ground tissue. Parenchymatous does ground tissue. Such type of arrangement of irregular arrangement of vascular bundles in the ground tissue is called atactostele. This is the spelling ATACTOSTLE. Atactostele. That means the ground tissue and vascular bundles occupies the central major part of the monocot stem. And each vascular bundle, as we have seen in the ground plan, the peripheral vascular bundles are smaller and central larger size. And each vascular bundle is enclosed with a sclerenchymatous tissue. Sclerenchymatous tissue. This sclerenchymatous tissue encloses. This is monocot vascular bundle. This is dicot vascular bundle. And few xylem elements are there. Xylem always towards the center and phloem always towards the periphery in each vascular bundle. And two metaxylem elements are clearly seen, which are large in size. They are metaxylem vessels. And one or two protoxylem elements also could be seen here. And one of the protoxylem element, you can see adjacent to it, there is a large air space. There is a empty space just adjacent to the protoxylem. That air space is due to is formed due to disintegration of protoxylem. Generally, it is having water or sometimes by air. That's why this is called air space, or air space. This is formed due to disintegration of protoxylem. That's why it is lysigenous cavity. This cavity. This is about vascular bundle. Let's have a look again. Yes, of monocot stem is having different parts. The first part is epidermis, second part is cortex, and the third part is the ground tissue. The fourth is vascular bundle. Epidermis, this is epidermis, single layered cells. Cells are barrel shaped, closely packed, without intercellular spaces. The external surface of epidermis is covered with thick cuticle. External surface, outer surface of the epidermis is covered with thick cuticle and no trichomes. This is about epidermis. You can find few stomata also in epidermis. The function of epidermis is protection and exchange of gases and prevents the transpiration. Because of the presence of stomata, it also helps in transpiration. Next, hypodermis. Generally, the cortex is reduced to hypodermis. Hypodermis is sclerenchymatous. It is few layered structure. Just below the epidermis, you can find sclerenchymatous hypodermis. Sclerenchymatous hypodermis. Few layered sclerenchymatous hypodermis is the key feature of monocot stem. It provides mechanical support. Rarely in some monocot stems you can find patches of chlorenchyma. Because of the presence of chlorenchyma, it photosynthesis. Generally, it provides mechanical support. And the third part of monocot stem is ground tissue. This is the ground tissue. It occupies large central column. The majority of the monocot stem is made up of ground tissue. The ground tissue is generally made up of parenchymatous tissue. These cells are parenchymatous cells, living, thin-walled, and loosely packed with small intercellular spaces. Loosely packed with small intercellular spaces are seen in the ground tissue. The main role of ground tissue is storage. It stores food materials. Generally, it is colorless. Cells are colorless. They don't have any chloroplasts. They have leucoplasts. They store food materials, and sometimes in some plants, these cells also store water. The main function of ground tissue is storage. And the fourth important part is vascular bundles. Here in monocot stem, the number of vascular bundles are numerous, countless. They are scattered irregularly in the ground tissue. Such type of arrangement of vascular bundles, the irregular arrangement, is called atactostele. In dicot stem, it is eustele. In monocot stem, it is atactostele. And each vascular bundle is enclosed with sclerenchymatous bundle sheath. The presence of sclerenchymatous bundle sheath is the key identification feature of monocot stem. And in each vascular bundle, you can see few xylem vessels and few phloem elements, which are side by side, which are together, and without vascular cambium. Hence, each vascular bundle is described as conjoint, collateral, closed type of vascular bundles. And the protoxylem is always in each vascular bundle is towards the center of the stem. That's why this condition is known as endark. Endo means inside. Whereas in root, the protoxylem is always towards the periphery. Such condition is termed as exarch in roots. But in stem, the protoxylem is towards the center of the stem, towards the inner side. That's why that condition is termed as endark condition. I'll repeat once again. In monocot stem, each vascular bundle is conjoint, collateral, closed type with endark condition. Here, one more important prominent feature of monocot stem is presence of lysigenous cavity. Lysigenous cavity or protoxylem lacunae. There is empty space adjacent to the protoxylem. It is filled with air or water. That cavity is formed due to disintegration of one of the protoxylem elements. That's why it is called protoxylem lacunae. If you see the differences between dicot stem and monocot stem, you can find a thin cuticle in dicot stem, thick cuticle in monocot stem. Trichomes are present in dicot stem. Trichomes are absent in monocot stem. Thin cuticle, dicot stem. Thick cuticle, monocot stem. Trichomes are present in dicot stem. Trichomes are absent in monocot stem. Very well developed cortex is there in dicot stem. In monocot stem, only the cortex is reduced to hypodermis. General cortex and endodermis are absent. General cortex and endodermis are absent. Only hypodermis is seen in monocots. And the central stele in dicot stem is made up of pericycle, vascular bundles, medulla and medullary rays. In monocot stem, no such structures are seen. Only here ground tissue and vascular bundles are seen. Large central column is composed of ground parenchymatous tissue. It contains many or numerous vascular bundles which are scattered irregularly. In dicot stem, is there. In monocot stem, atactostele is there. U-stele is a stele where the limited number of vascular bundles are there, which are arranged in a circle. Arranged in a circle in dicot stem. Whereas in monocot stem, the number of vascular bundles are numerous. They are scattered irregularly. There is no particular fashion of arrangement of vascular bundles. They are scattered zigzagly, scattered over in the ground tissue, that is parenchymatous tissue. There is no particular fashion of arrangement of vascular bundles in monocot stem. Such type of vascular bundles or arrangement of vascular bundles is termed as atactostele. In dicot stem, vascular bundles are conjoint, collateral, open type condition. Open endark condition. Whereas in dicot stem, endark condition. In monocot stem also endark condition. In dicot stem, vascular bundles are conjoint. Here also conjoint. There also collateral. Here also collateral. But there is a slight difference. In dicot stem, they are open type. In monocot stem, they are closed type. What do you mean by open and closed? There is a vascular KMBM is present between xylem and phloem. A strip of KMBM, a strip of meristematic tissue is present between xylem and phloem. Such type of vascular bundle is known as open type. Whereas in monocot stem, you cannot see such a KMBM in vascular bundle. A strip of KMBM is absent. They are together. Xylem and phloem are together in monocots. That's why they are called closed type of vascular bundles. In both the stems, you can see the endar condition, condition, but there is no protoxylem, or lysogenous cavity in dicot stem. But this lysogenous cavity is seen in monocot stem. Pericycle is present in dicot stem, absent in monocot stem. Medulla, medullary rays are clearly seen in dicot stem. Here, only ground tissue is seen in monocot stem. Whereas in dicot stem, there is no bundle sheath. Here in monocot stem, there is a sclerenchymatous bundle sheath is there. In that, see a fiber-like structures in sugarcane also. When you extract the juice from the sugarcane stem, you can find a string-like structures. Those are nothing but they are vascular bundles which are enclosed with sclerenchymatous fibers. A pulp which is seen in the sugarcane stem, that is pulp is nothing but that is simple parenchymatous tissue. Simple parenchymatous tissue. These appear like threads or fibers in the sugarcane stem and sorghum stem. Even in bamboo also you can find such strips. Fiber-like strips can be seen along the length of the stem. Those are nothing but they are vascular bundles which are enclosed with sclerenchymatous fibers. Okay, with this, let me conclude. Tomorrow, we'll discuss internal structure of leaf, that is anatomy of leaf. Okay, thank you. Let me conclude, stop here. So this is also important for examination. So out of 4 transverse sections, 2 are roots and 2 are stems. These 4 are very, very important for examination point of view. You need to practice the figures. You need to draw the figure. If you get essay on monocot TS, you need to draw this diagram along with vascular bundle. This vascular bundle. Sector and vascular bundle are enough. And you have to label. You have to label the sector, enlarged portion, this enlarged portion, and vascular bundle. This enlarged portion and vascular bundle you need to draw. You have to practice. This is called enlarged portion. And this individual, if possible, this individual vascular bundle also you need to draw. And you have to label. And you have to describe under 4 different subheadings. And you have to practice the differences also. You have to read. And you have to read some tips. Use steel. Definitions are there. Use steel and attack to steel. The differences also you need to know. So with this, let me conclude. All the best.